So now we'll start. Good morning, students. And today we are speaking about the tissues of internal environment, the new group of tissues. We'll continue to speak about the tissues. And now the tissues of internal environment. This slide presents the main functions of tissues of internal environment. And the first function is to provide the structural and metabolic support for other tissues and organs of our organism. Second function of the tissues of internal environment is that mediate the exchange of nutrients, metabolites, waste products between the tissues and circulatory system. And the third function of tissues of internal environment is the protection of our organism and a formation, a site for the storage of fat. Look at the classification of the tissues of internal environment, and this group of tissues is classified into the blood and lymph, into the connective tissue proper, which is subdivided into the fibrous connective tissues, loose and dense, skeletal connective tissues with special properties, and embryonic tissues. So today we are speaking about the blood and lymph and about the processes of formation of blood, processes of hemopoiesis. Next class, we'll speak about the connective tissue proper. Next lecture, we'll speak about the connective tissue proper, connective tissue skeletal, of the skeleton, and other types of connective tissues. Look at the main features, main distinctive properties of tissues of internal environment. The first property, predominance of extracellular matrix over the cells. Large volume of extracellular matrix we can see in the tissues of internal environment. Second main feature, second main characteristic, cells of these tissues lie separate from one another. There is large distance between the cells in the tissues of internal environment. And the last feature, last characteristic of these tissues is the development from mesenchyme. What is mesenchyme? Look at this. Mesenchyme is a kind of embryonic tissues. Mesenchyme originates from middle embryonic layer, mesoderm, and this layer forms the multipotential cells named mesenchyme. Look at this picture. Mesenchymal cells have long processes, do not create the intercellular contacts with each other, and migrate throughout the bodies of the embryo to form the different kinds of tissues. So mesenchyme is the main source of embryonic development of all tissues of internal environment. So now we are speaking about the blood, the first tissue of internal environment. Blood is a part of blood system. Blood system includes two main portions: blood as a tissue and organs of hemopoiesis, where the blood, the main parts of the blood are formed. About the organs of hemopoiesis, such as bone marrow, such as lymph node, spleen, thymus, we'll speak next semester. But now we are speaking about the structure of the blood. Look at the main functions of the blood. The first large function is transport function. This common function includes some small functions, such as trophical function. What does it mean? The structures of the blood transport the different nutrients, and this function is named trophical function. Next small function is called respiratory function. What is it? The formed elements of the blood transport the gases, oxygen and carboxygen. So this function is named respiratory function. Next function, formed elements of the blood transport the hormones and other biological active substances which regulate the functional activity of all organs, and this function is named regulatory function. And the last part of this large function, formed elements of the blood transport the metabolic products which leave our organism, transport these products outside our organism, and this function is named excretory function. So the first large function of the blood is named transport of different substances. Next function, next large function of the blood is named the homeostatic function. What does it mean? The formed elements of the blood and plasma of the blood participates in maintaining the constancy of internal environment of our organism, participates in the storage of constancy of our inner organs. So this large function includes the regulation of osmotic balance, acid-base balance, body temperature, and another functions of our organism. And the last function of the blood is the defending function. Blood provides the different types of immune defense of our organism. Blood is responsible for the coagulation of blood coagulation processes and other protective functions. So these are main functions of the blood, and look at the main components of the blood. The main components, composition of the blood. So blood is represented by two main parts. The first part is named plasma. Plasma is a modified extracellular matrix, extracellular substance, and plasma, the composition of plasma from 55 to 60% from all volume of blood. Second part of the blood, second component is formed elements. Formed elements is a group which includes cells and cells derivatives. These are the postcellular formations, postcellular structures without nuclei. So formed elements from 40 to 45% of all volume of blood. So now look at the main percentage, and now we are speaking about the blood plasma. Look at this. The chemical composition of blood plasma water up to 91 to 92%, and different organic and non-organic substances. Organic substances such as plasma proteins, such as plasma lipids, and different carbohydrates. And non-organic substances, it's a mineral salts, different microelements up to 1 to 2%. What about the plasma proteins? From 7 to 8% of proteins we can see in the plasma, and all plasma proteins are subdivided into the three main groups: albumins, globulins, and fibrinogens. Albumins, the first plasma proteins, about 60% of plasma proteins, play a great role in maintaining oncotic blood pressure. Albumins connect with the water, attach the water molecules, and storage the water inside the blood vessels. This feature is named keep the oncotic pressure of the blood. Second main function of albumins is transport. Albumins transport such biological active substances as and chemical substances as bilirubin, cholesterol, different acids, bile acids, and also drugs. Next protein is named globulin. So globulins from 20 to 30% of all proteins of the blood are subdivided into the three main groups: alpha globulins, beta globulins, and gamma globulins. Gamma globulins are the antibodies, or in other names immunoglobulins, takes part into the immunological defense of our organism against different alien substances named antigens. Alpha and beta globulins takes part in the transport of different chemical substances such as lipids, hormones, vitamins, and microelements. And fibrinogen, from 4 to 8% of all plasma proteins, is the protein which is necessary for the blood coagulation. Fibrinogen transforms into the protein fibrin and takes part into the formation of thromba. So blood plasma is a kind of extracellular matrix. And now we'll start to speak about the formed elements of the blood. Formed elements of the blood includes cells, leukocytes, and postcellular formations, structures without nuclei, such as erythrocytes and platelets or thrombocytes. So formed elements are subdivided into the three main types: erythrocytes, leukocytes, and platelets, or another name thrombocytes. And look at this slide. This slide shows the total counts of formed elements per 1 liter of blood. Total counts of formed elements in 1 liter of blood is named blood formula. These total counts you should know by heart. Another name of blood formula is hemogramma. This is the blood analyzed, and your future patients show this blood formula to you every day. So erythrocytes in the blood formula, the counts of erythrocytes is differ for men and women. In man organism, count of erythrocytes 3.9 to 5.5, 10 raised to 12 in a liter. For women, 3.7 to 4.9, 10 raised to 12 in a liter for women. Leukocytes, the main the normal composition, from 4 to 9, 10 raised to 9 in a liter, and thrombocytes, 180, 320, 10 raised to 9 in a liter. So these are normal counts of font elements per 1 liter of the blood. So look at this blood formula. Prevalent population of formed elements is population of erythrocytes. Erythrocytes, 10 raised to 12 in a liter. And now we'll start to speak about the erythrocytes. We'll start to speak about the erythrocytes. Look at these illustrations, electron micrograph, light microscopes we can see, and the main function of erythrocytes is transport of gases, oxygen, carboxygen. It is a respiratory function. Also, the main function of erythrocytes is the transport of hormones, transport of drugs, absorb of the surface drugs, hormones, different toxins also can absorb on the surface of the erythrocytes. Amino acids, immunoglobulins are also transported by the erythrocytes. In normal condition, the erythrocytes, according to the sizes, are subdivided into the 3 types. Normocytes, the diameter from 7 to 8 micrometers. Macrocytes, diameter more than 8 micrometers. And microcytes, diameter less than 6 micrometers. A change in the size of erythrocytes occurs in diseases and named anisocytosis. Anisocytosis is abnormal size of erythrocytes. In normal condition, the prevalent kind of erythrocytes is normocytes, 75% normocytes, macrocytes 12%, and microcytes 12% only. And the changes in this proportion is named anisocytosis. Look at this also illustration. Look at this electron micrograph. The most erythrocytes have biconcave shape. This shape is named disc-shaped. Looks like discs. The central part of erythrocytes is thinner than the peripheral portions. That is why if we use the light microscope, we can see light central, look like the white spot, and dark red peripheral part, because peripheral part is thicker than the center, disc-shaped cells. But some erythrocytes are spherical in shape, for example, and named spherocytes. these erythrocytes are named discocytes. Spherical erythrocytes are named spherocytes, for example. Sometimes the erythrocytes have small projections. These erythrocytes are named echinocytes, but mostly erythrocytes are disc-shaped. The next moment, poikilocytosis, a normal condition, is a prevalence of abnormally shaped erythrocytes. Prevalence of abnormally shaped erythrocytes. So what can we tell about the structure of erythrocytes? Erythrocytes are not the cells because all erythrocytes don't contain the nuclei. Nuclei are absent. The main quantity of organelles also are absent, and cytoplasm of erythrocyte is filled by the pigment named hemoglobin. Look at this. Hemoglobin is a substance, complex chemical substance, consists of two main parts, as you know. Hema is the serum-containing part, and globin is a protein-containing part. Students, where are the another students? Only 13 students are present in the lecture. What about another students?
Профессор, я все сказали, и в группе тоже написал, и вы зачем не будет и на занятии. Я все сказал.
Please make me the screenshot of students who are present of our lecture and pass me in WhatsApp.
Хорошо, хорошо.
So I'm waiting after the lecture. Please pass me the students who are present on here. Tell me, everything is okay? Sound, presentation?
Yes.
Yes, professor, yes.
Yes? Mm-hmm. Okay. So we are speaking about the structure of erythrocytes, and I told that erythrocytes don't contain nucleus, don't contain the main quantity of organelles, and houses the pigment hemoglobin, which connect and which contact with oxygen and carboxygen. Hemoglobin consists of two main parts, hema, serum-containing part, and globin, protein-containing part. There are two main types of hemoglobin in the human organism. Hemoglobin A, or adult hemoglobin, and hemoglobin F, fetal hemoglobin. In adult person, the hemoglobin A is prevalent, up to 98%, and only 2% hemoglobin F. In newborns, for example, hemoglobin F prevalent up to 80%, and only 20% hemoglobin A. So now we are speaking about the plasmalemma of erythrocytes. Plasmalemma of erythrocytes have ability to not absorption, but attaching the substances for the transport. For example, plasmalemma absorb the hormones, drugs, absorb the immunoglobulins, amino acids, and transport these substances inside the circulatory system, transport these substances to the other tissues and organs. So, as you know, erythrocytes can change their shape. Usually, erythrocytes are disc-shaped, but in some cases, the diameter of blood vessels, for example, diameter of blood capillaries, less than the diameter of erythrocytes. In this case, in this situation, erythrocytes change their shape. So it's possible because plasmalemma, cell membrane of erythrocytes, has ability to change. Plasmalemma is flexibility, strength. So that is why erythrocytes can change their shape. There are many proteins are present in the plasmalemma of erythrocytes. These are transport proteins, transport canals. These are receptor proteins. So proteins of blood groups also presence in the plasmalemma of erythrocytes. And another. So more than 15 proteins are present in plasmalemma, but the main proteins are present in these slides. The names of these proteins are glycopharin, ankyrin, protein 3, protein 4.1. So these are the main proteins of plasmalemma. Look at the glycopharin. Glycopharin is a transmembrane protein which attach the carbohydrates. Attach the carbohydrates. We can see the change of carbohydrates on the surface of these proteins, and the main function of this protein is receptor function, recognize the different substances. Next, protein transmembrane, also protein 3. This protein is necessary for the exchange of carboxygen and oxygen, so gas exchanges. Next, protein 4.1, unusual name 4.1, connects with the elements of cytoskeleton and actin filaments, and also connect with peripheral protein spectrin. So this protein, this complex of protein, help to change the shape of erythrocyte if it is necessary. So we are speaking about the structure of erythrocyte. The lifespan of erythrocyte is 120 days, and the precursors of erythrocytes are named reticulocytes. Reticulocytes, precursors, younger erythrocytes, and look at the main feature of erythrocytes. Of reticulocytes, in the cytoplasm of reticulocytes, we can see the remains of organelles. Erythrocytes do not contain the organelles, but reticulocytes in the cytoplasm, if we use specific method of staining, we can see the remains of different organelles. In normal condition, in peripheral blood, we can see up to 1% reticulocyte in relation to the total number of erythrocytes. The increase of the content of reticulocyte is observed with bleeding, blood bleeding, and hemolytic anemia. So the increasing of percentage of reticulocyte we can meet in anemia and blood bleeding. So that's all about the red blood cells, red blood elements, erythrocytes. And now we are speaking about the next formed element of the blood, named platelets, or another name, thrombocytes. So the main function of thrombocytes is participation in the blood coagulation, formation of thromba, and secrete some biological active substances that are involved into the repair of blood vessels. The lifespan of platelets is from 7 to 8 days only, and the precursors of platelets are the cells, large cells with large volume of cytoplasm, named megakaryocytes. Megakaryocytes are situated in the bone marrow. These are polyploid cells with large volume of cytoplasm and many organelles, and these cells, megakaryocyte, situate near the blood vessels and near the blood capillaries of the bone marrow, and release the small portions of their own cytoplasm to the blood stream. And these small portions are named platelets. Unusual development of these formed elements, and we can see the unusual plan of structure. So look at the slide, morphological and structural organization of the platelets. Platelets or thrombocytes have no nuclei and have no most organelles. We can see only the remains of organelles. In the platelet, we differ two main portions. Hyalomere, look at this number first, which is situated on the periphery of the platelet, and second, granulomere. Granulomere is situated mostly in the center of the platelet. We'll start to speak with the hyalomere, which is located on the periphery of the blood of the thrombocyte. So granulomere is represented by two parts. First part is the elements of cytoskeleton of platelet, and the second part in the system of canaliculi, open and dense canalicular system, or another name, tubular system. This is the same name of canalicular system. So look at this. Plasmalemma covers the platelet. Plasmalemma is characterized by the large volume of glycocalyx, thick layer of glycocalyx. There are numerous receptors situated inside the glycocalyx. Glycocalyx is necessary for the adhesion of platelets to the vascular wall and aggregation of thrombocytes together to form the thromba, if it is necessary for the coagulation of blood. So plasmalemma with thick layer of glycocalyx, and below the plasmalemma there are many elements of cytoskeleton. Which elements of cytoskeleton? Concentric rings of microtubules. From 4 to 15 microtubules form the bundles, and these concentric rings of microtubules situates on the periphery to form the hyalomere of thrombocytes. Below the microtubules, there are the microfilaments, also elements of cytoskeleton. Microfilaments are made by the contractile proteins such as actin, myosin, gelzalin. These contractile proteins involved into the contraction of platelets and retraction of thromba. So it is a hyalomere. It is the first portion of hyalomere. elements of cytoskeletal. And the second portion of hyalomere is a canalicular system, open and dense canalicular system. Open
fact that this is reality, I'm not concerned about had it been the other way around, how it would have been adjusted or adjudicated. But had it been a routine clash, would it have had this level of scrutiny? And is that the place we now find ourselves in terms of how, especially games with Real Madrid, Barcelona, and let's extend Atletico Madrid, how are they managed from a refereeing perspective in comparison to all the other games in Spanish football?
1 minute wait and then we'll continue our lecture.
Хорошо, профессор.
So now we'll continue. We'll speak about the structure of thrombocytes, so another name, platelets. And I told that there are two main parts in each platelet. On periphery, there is hyalomere, and in the center, there is granular mere. Hyalomere is represented by the cytoskeleton, elements of the cytoskeleton of the thrombocyte. And hyalomere mostly performs the supporting function. On the periphery, we can see the rings of microtubules, from 4 to 15 concentric rings. And below the microtubules, there are the microfilaments made by the contractile proteins such as myosin, actin, gilzalin, which take part in the retraction of thrombum. Thrombus becomes dense during the retraction. And also hyalomere is represented by system of canaliculi, open canalicular system and dense canalicular system. Open canalicular system is the invaginations of plasma lemma of thrombocytes, and open canalicular system is necessary for the transport of different substances, absorb the substances inside the cytoplasm of thrombocyte and excrete the substances outside. And dense tubular or dense canalicular system is the slim tubules inside the platelet, slim tubules, which storage the calcium ions and also produce the system of tubules prostaglandins. Prostaglandins are the biological active substances which are necessary for the processes of blood coagulation. So we told about the hyalomere, and now we are speaking about the granular mere. Granular mere situates in the center of thrombocyte and consists of system of granules. All granules are subdivided into the three main groups: alpha granules, delta granules, and gamma granules. All granules houses biological active substances with many important functions. For example, alpha granules contain the glycoproteins such as fibronectin and fibrinogen. These proteins necessary for the blood coagulation and formation of fibrin and thromba. Also, alpha granules houses the thrombospondin, also necessary for the formation of thrombus, and another factors of blood coagulation. Delta granules houses another substances, calcium ions. Without calcium, blood coagulation does not take place, and also adenosine 3-phosphate acid, adenosine D-phosphate acid, in production of energy and energetic substances. And also serotonin and histamine, all necessary for the formation of thrombus. And the smallest granules of platelet named lambda granules. Lambda granules are the sort of lysosomes. And these granules houses the thrombolytic enzymes for the lysis of thromba and another biological active substances, different lysosomal enzymes, acidic hydrolases, lipases, phosphorylases, phosphatases, and another lysosomal enzymes. So granules are present in granular mirror, and also some remains of organelles such as mitochondria, such as ribosomes, the short cisterns of rough endoplasmic reticulum, elements of Golgi complex, glycogen inclusions are also present in the granular mirror. And now we told about the erythrocytes. I repeat, we told about the platelets or thrombocytes. And now we are speaking about the largest elements of the blood, largest group of formed elements of the blood, named group of leukocytes. So look at the leukocytes, and at first I'll tell you that all leukocytes house the nuclei in their cytoplasm. All leukocytes have ability to active movement, leave the blood vessels and migrate to the connective tissues, perform their functions mostly in the connective tissues. This is a morphological classification of leukocytes and also leukocytorial formula. Leukocytorial formula is a percentage of different kinds of leukocytes to the general quantity of leukocytes. So the first classification. According to the presence of the granules in the cytoplasm, all leukocytes are subdivided into the agranulocytes without granules and granulocytes with the specific or granules in the cytoplasm. Usually agranulocytes is characterized by the presence non-segmented nuclei. Granulocytes have segmented nuclei in their cytoplasm. All types of leukocytes are characterized by the presence of primary non-specific granules in their cytoplasm. Non-specific granules are named primary granules, other lysosomes. And these lysosomes houses the lysosomal enzymes, antimicrobial proteins, and another biological active substances. But granulocytes houses the secondary granules, specific granules in the cytoplasm. And according to the affinity to dyes, according to the staining of these granules, all granulocytes are subdivided into the eosinophils. They are granules stained by acid dyes into the pink color or red color. Only 0.5 to 5% of eosinophils in the formula. Basophils, these specific granules are stained by the basic dyes into the blue or purple color. And the neutrophils. Neutrophils, they are granules in the cytoplasm stained both by acid and basic dyes. Neutrophils, the normal condition in normal blood, from 48 to 78%. Basophil from 0 to 1% only. So specific granules in the cytoplasm houses the specific biological active substances which are responsible for the functions of each type of granular cyte. And one more classification of neutrophils according to their age. All neutrophils are subdivided into the young neutrophils, into the band neutrophils, and mature neutrophils. Percentage on different types of neutrophils you should know also by heart. All these percent you should know by heart because it's leukocytoral formula, it's a blood analyzer of your future patients. And granulocytes do not have the specific granules in the cytoplasm, and granulocytes are subdivided into the lymphocytes and monocytes. All granulocytes have ability to phagocytosis. Phagocytosis is the digestion of different alien substances. These alien substances, bacteria, viruses, toxins, are named antigens. Different alien substances named antigens. All granulocytes have ability to phagocytosis. And now we'll speak about the first type of granulocytes named neutrophils. And I repeat that neutrophils are subdivided according to their age to the young, bands, and mature. The mature is the presence of mature neutrophils is larger than the presence in other types of neutrophils in peripheral bloods. So look at the neutrophils, which are the main morphological characteristics. All neutrophils, mostly mature neutrophils, are characterized by the segmented nucleus. We can see from 2 to 5 segments in one nucleus, which connect with each other by the bridges, from 5 to up to 5 segments. And cytoplasm is filled by the small, weakly staining granules, and these granules are stained both by acid and basic dyes, such as why these cells are named neutrophils. Some specific substances are present inside the granules. So these granules houses them such substances named lysozyme. Lysozyme acts as antibacterial substance and destroys the bacterial wall. Also, these granules houses the lactoferrin. Lactoferrin causes the adhesion of bacterial cells. Also, these granules houses the complement activation, different enzymes which causes the lysis of bacterial cells. So all these biological active substances act as the antibacterial substances and necessary for the antibacterial protection. That is why the main function of neutrophils is the protection against the bacteria invasion, against the pathogenic act of bacteria. So look at this table. Phagocytosis of bacteria and antibacterial functions are the main functions of neutrophils. Bacteriolysis, bacteriostatic function, and phagocytosis of bacteria. These are the substances inside the specific granules of neutrophil. If we use if we see the bacterial inflammation in the organism of our patient, we can see the increase of percentage of neutrophils in the leukocytorial formula. So mature neutrophils are characterized by segmented nucleus, but young and band neutrophils have non-segmented nucleus. Usually band neutrophils have a nucleus which looks like a stick, and the young neutrophils nucleus has bean-shaped nucleus, another types of nucleus. So in leukocytorial formula, we can see the increase of young and bands neutrophils in some cases, in some pathological condition, and this picture is called left displacement of left shift of leukocytorial formula. It is increase of young and band neutrophils and decrease of mature neutrophils. Left shift of leukocytorial formula we can meet during the bacterial inflammation in the organism of our patients. Bacterial inflammations. So neutrophils, segmented nucleus, and specific granules with antibacterial substances. Next granulocyte is named eosinophil. Look at the morphological characteristic of eosinophil. Usually we can meet two segments in the nucleus connect by the bridge, bisegmented nucleus, and cytoplasm is filled by granules, and these granules stained by acid dyes into the red color or pink color, red or pink color. So these specific granules houses such biological active substances as eosinophilic cationic protein, eosinophilic neurotoxin, histaminase, peroxidase, and the main basic protein. So granules on the electron micrograph is characterized by the presence of the dense central part, dark centers we can see in these granules. According to the chemical composition of the granules, eosinophils takes part in allergic inflammation. So by these substances, eosinophils decrease the early GIR and also destroys the parasites. The main function is an anti-parasitic function, protection against the parasites, and also decrease the allergic inflammation. So these are the eosinophils. Neutrophils and eosinophils have the same lifespan from 7 to 8 days only. And the last granulocyte is called the basophil. Look at the morphological features of basophils. Basophil is characterized by weakly segmented nucleus, and the cytoplasm is filled by large granules which are stained metachromatically. What does it mean, metachromatically? Metachromasia is a ability to changes the color of dyes. For example, if we use blue dyes, granules are stained into the black color. Or, for example, if we use blue dyes, granules are stained into the dark red color. So change the color of dye as metachromasia. So granules of basophils house such biological active substances as heparin, histamine, prostaglandins, serotonin, and leukotrienes. And the main function of basophils is to increase the blood flow, increase the vascular permeability, and mobilize the rest of granulocyte into the place of damaged. So, in common, the main function of basophils is regulations of homeostasis of the tissues. Regulation of homeostasis of the tissues. The lifespan of basophils is longer than the lifespans of another types of granulocytes, from 1 to 2 years. So we told about the group of granulocytes and about the morphology and their functions. And now we are speaking about the group of agranulocytes. These leukocytes have no specific granules in the cytoplasm, and their nuclei are non-segmented nuclei. So we'll start to speak about the lymphocytes. Look at this. The nucleus of lymphocyte is spherical in shaped, round in shaped, and slim ring of cytoplasm surrounds this nuclei. All lymphocytes are subdivided according to their sizes into the larger or activated lymphocytes and small inactive lymphocytes. Diameter of large lymphocytes more than 11 micrometers. Diameter of small lymphocytes approximately 5 to 7 micrometers. And also we can differ middle lymphocytes also in normal condition. One more classification of lymphocytes. It's a classification according to their functions and according to their place of origin. According to their place of origin and functionally, all lymphocytes are subdivided into the T lymphocytes, B lymphocytes, and 0 lymphocytes. The place of origin of T lymphocytes, one of the stage of their differentiation is thymus. That is, thymus is the central organ of hematopoiesis, and that is why these lymphocytes are named T lymphocytes. In the thymus, the effector immunocompetent subpopulations of T lymphocytes are formed. Immunocompetent subpopulations are present here in this table. All T lymphocytes, according to their functions, according to the receptors on their surface plasma limb, are subdivided into the T helpers, T suppressors, T cytotoxic cells, and T memory cells. T cytotoxic cells, what about the functions? T cytotoxic cells destroy their genetically alien cells. For example, cells of tumors, cells of transplanted, or another genetically alien cells, destroy them. T helpers and T suppressors regulates their immune response to the invasion of different foreign substances. B lymphocytes. B lymphocytes originates into the bone marrow. some stage of differentiation of these lymphocytes take place in the bone marrow. That is why these lymphocytes are named B lymphocytes. B lymphocytes, a factor of immunocompetent subpopulations, are present in this table, and B lymphocytes are subdivided into the plasma cells and B memory cells. Plasma cells are the cells which produce the antibodies or immunoglobulins that react with alien substances named antigens, react and kill these alien substances named antigens. So plasma cells are the sort of B lymphocytes. T memory and B memory cells storage the informations about the alien substances, about the pathogens, and form the immediate immune response during the second or third invasion of these antigens. And zero lymphocytes, or natural killers, also kill the different alien substances, different pathogens. So this is the classification of lymphocytes according to their development, according to the source of their origin, and according to their function. Percentage of lymphocyte types to the total number of lymphocytes are present in this table. Prevalent population is population of T lymphocytes up to 80%, B lymphocyte up to from 8 to 20%, and natural killers in the blood from 5 to 20%. The short characteristic of the functions of lymphocytes are present here. There are two types of immune protection of our organism: cell-mediated immune protection or adaptive immunity, and humoral immune protection. Cell-mediated immune protection means recognition and killing the foreign cells. And cell-mediated immune protection is realized by T cytotoxic lymphocytes and also natural killers. And second type of immune protection is humoral immune protection, is a recognition of foreign substances, foreign antigens, and production the antibodies which bind the antigens and destroy the antigens. Humoral immune protection is performed by the plasma cells producing the antibodies or immunoglobulins. And this slide shows the characteristics of humoral immunity and cell-mediated immunity. I repeat because it's difficult. Humoral immunity is mediated by the antibodies or immunoglobulins that act directly on antigens. These antibodies are produced by plasma cells derived from B lymphocytes. And cell-mediated immunity, cell-mediated immune protection is mediated by T cytotoxic cells that attack and destroy the host of foreign cells, virus-infected cells, cells of tumors, cells of transplantat. So cell-mediated immune response is created by cytotoxic T lymphocytes and also natural killers. And the last type of leukocytes, granulocytes, also are monocytes. Monocytes, the normal condition percentage of monocytes, look at this, from 3 to 11%. From 3 to 11%. Look at the morphology of monocyte. morphology of monocyte. These are the largest leukocytes. Their size up to 20 micrometers, largest cells in the blood. Look at this. This type of monocyte, this type of leukocyte is characterized by the presence of bean-shaped nucleus, and large volume of cytoplasm with all common organelles. Granules are not present. This is a granulocyte. Monocyte situates in the bloodstream only during the several hours and quickly migrate through the blood vessel wall to the connective tissues. In the connective tissues, monocyte transform into the macrophages, cells of connective tissue, and perform their main functions. The main functions of macrophages are the phagocytosis of different alien substances, different pathogens, production of some bactericidal substances, bacteriostatic, bacteriolytic substances, and also takes part into the immune reaction, act as a antigen-presenting cells. We'll speak about this group of cells next semester when we'll speak about the immune response and when we'll speak about the organs of hemopoiesis and also immune system. On your practice class, you should look into the onto the blood smear, and you should recognize the different kinds of formed elements in the blood according to their morphology. Look at this. For example, bisegmented nucleus and pink granules in the cytoplasm are the main characteristics of eosinophils. Large spherical nucleus and small slim ring of cytoplasm around this spherical nucleus is a lymphocyte. And also you should recognize another types of leukocyte into the blood. And short information, because we have only 7 minutes, short information about the hemopoiesis. Hemopoiesis, it is a process of formation and regeneration of formed elements in the blood. According to the date, according to the age of this formation, hemopoiesis are subdivided into two periods: prenatal or embryonic hemopoiesis and postnatal or post-embryonic hemopoiesis. Prenatal hemopoiesis is a histogenesis of blood as a tissue and regeneration of blood, renewal of blood-formed elements and replacement of blood-formed elements. In post-embryonic hemopoiesis, we can see only the regeneration of blood and not the formation of blood as a tissue. Blood regeneration includes also the renewal of blood-formed elements and replacement of blood-formed elements. In embryonic or prenatal hemopoiesis, we differ three stages takes place in different weeks of embryonic period. The first stage is named mesoblastic, and on this stage takes part from 2 to 9 weeks of embryogenesis. The formation of blood takes place outside the embryo in the extra-embryonic organs such as yolk sac and chorion and also amnion. On the second and third periods of embryonic hemopoiesis, formation of formed elements of the blood takes place inside the embryo, in different organs of the embryo. These stages are named hepatolenal stage from 5th to 14 weeks of embryonic period. On this stage, formation of blood takes place in the liver, in the thymus, in the lymph nodes, and in the spleen. And the third stage of embryonic hemopoiesis is named thymico-medullary hemopoiesis. On this stage starts from 10 weeks of embryogenesis and lasts up to 14 weeks of embryogenesis. On this stage, formation of blood takes part in red bone marrow and continues in the thymus. 3 stages in embryonic hemopoiesis. In post-embryonic hemopoiesis, in post-embryonic period of hemopoiesis, regeneration of blood takes place in mostly two groups of organs. Central organs, including bone marrow and thymus, and peripheral organs of hemopoiesis, such as lymph node, such as spleen, appendix, tonsils, and lymphoid follicles in the digestive and respiratory system. So, postnatal hemopoiesis is a formation of new formed elements, and we can see the structure of hemopoietic differon, how the precursors of formed element transform into the mature formed element, into the specialized formed element of the blood. Hemopoietic differon includes 6 classes of cells. First class is named hemopoietic stem cell. These are the precursors of formed elements, which can differentiate to any formed elements of the blood. These cells are named pluripotential. Their pluripotential cells can transform into the any formed elements of the blood. Second stage of differentiation and second class is named multipotential progenitor. Multipotential progenitor are subdivided into the precursors of myeloid cells and precursors of lymphoid cells. Myeloid precursors developed to form the granulocytes, erythrocytes, monocytes, and thrombocytes, and lymphoid precursors transform only into the lymphocytes. Third class is called unipotent precursor. Unipotent precursor transform only in concrete one type of formed element, unipotent precursor. For example, precursor of erythrocytes, precursor of monocyte, and another unipotent precursors. Fourth class is called blasts. For example, erythroblasts, monoblasts, myeloblasts, lymphoblasts, and another types precursors. The fifth class is named maturing cells. For example, these are the maturing precursors of erythrocytes and maturing precursors of granulocytes. And the last class are named specialized cells. And about the specialized cells, we told all this lecture. That's all for today. See you week after next. We'll continue to speak about the tissues of internal environment.